Thank you for the introduction, and thank you to the organizing committee for the opportunity to speak here today. It's a distinct honor to be speaking, particularly at the plenary session at the SID, as I've been attending this meeting since I was in medical school, working with Steve Katz as a Howard Hughes Scholar at the NIH. It's bittersweet for me to speak today, particularly since the session is in honor of Steve. We all know that he's just been an incredible force in dermatology and skin biology throughout the world. He's certainly had a tremendous influence on my life and career. So much so, he was so good at this that he actually made me feel special until I realized he has this effect on everybody and many individuals in this room. And I think that my only regret is that he's not here with us here today. I have a number of disclosures, mostly arising from the work, some of the work that I'll be speaking about here today. So a central focus of our research program has been trying to understand a very basic process in the skin. And that is really understanding how healthy skin becomes diseased or inflamed skin. So how do we get a rash? And what are the key initiating events that actually trigger this process? As a dermatologist, this is a very simple but profound question for us to pursue. We're also interested in understanding how, if we get inflammation in the skin, how other pathologic processes set in, such as itch. When I worked with Steve as a medical student, I first became very interested in understanding how the epithelial barrier itself can speak to the immune system to engage the immune system. We were studying this in the context of graft versus host disease. But this concept of epithelial immune crosstalk stayed with me. And at around this time, there was a early source of inspiration, which was this work by Irwin McLean. The identification of filaggrin mutations at the skin barrier predisposing to the development of atopic dermatitis, in my mind, was a major conceptual advance. It solidified in my mind the notion that the barrier is not just simply a barrier, but it actually has a very important role in regulating the immune system, particularly in the context of atopic dermatitis and allergic diseases. When we first were studying atopic dermatitis or eczema, therapeutic options were woefully limited, despite the fact that this condition was so prevalent and had a high morbidity. I think this is rapidly changing before our eyes with the development of new therapeutics now. And the paradigm was relatively straightforward at that time, and it was that if you have a healthy skin barrier that's somehow perturbed, perhaps due to filaggrin deficiency, we get inflammatory stimuli or allergens that somehow translocate this leaky barrier and activate the adaptive Th2 cell, which can recognize the antigen, but is characterized by the production of the Th2 cell-associated cytokines IL-4, 5, and 13. This actually results in a variety of late-phase effector events, including activation of mast cells, which are coded with IgE. We get elevated production of IgE, which is a defining feature of atopic dermatitis and atopy in general. And as well as by work by Lisa Beck and Donald Leung, that these cytokines actually directly damage the barrier itself and can even further downregulate expression of filaggrin. But there was actually a very interesting development around this time as well, which was the identification of epithelial cell-derived cytokines like IL-25, IL-33, and TSLP. These cytokines were known to be produced by stressed or damaged epithelia at multiple barrier surfaces, not just the skin, and would broadly activate this pathway. When we actually delved into what the mechanisms were, we had some unexpected turns, including the identification that circulating basophils get it recruited into the skin and are a major source of IL-4. Furthermore, that previously unrecognized group 2 innate lymphoid cells, or ILC2s, were present in the skin and that are major sources of IL-5 and IL-13. These cells very much resembling T cells but lacking antigen-specific T cell receptors. So the first work, our foray into this field was this, along with other work that we contributed to here. A number of other studies have further expanded upon this biology, and this has been very much validated in a variety of different models, both in mice as well as in human biology as well. So at this point, we were interested in understanding how these innate cell populations actually drive the development of inflammation and rash. But then in recent years, we've actually turned our attention to what actually happens when we have inflammation in the skin and how pathologic itch develops. This group of people are not meant to be comprehensive. There are many others who have contributed to this, but these are individuals that have greatly influenced our work based on major contributions to understanding of clinical itch over the decades. I think really good clinical insight has actually provoked a number of hypotheses that have become increasingly testable in the laboratory, and for that we're grateful to these individuals for their clinical advances. On the basic science end, basic advances in itch biology, again not meant to be comprehensive, but highlights of various molecular as well as cellular discoveries in itch biology have emboldened us and others to really go forward to try to understand itch at a very basic level. These are some of the individuals that have been involved in these studies. What these studies really did was that help us to understand that itch is not simply some mild form of pain or a non-essential pathway, but these are can be very tractable at the molecular level. So when I think about itch, I like to keep it really simple. Something we can all relate to. We all know what histamine is. Histamine is actually a classical pruritogen. It has a number of other functions, but if we inject histamine to the skin, we can evoke itch. That's because histamine can bind its receptor on the sensory neuron, and by way of its dependence on cation channels like TRPV1, can actually eventually activate an action potential that then goes to the spinal cord, works its way up into the brain, and we perceive itch. I'll come back to the concept of histamine later on in the talk, but also recent work has identified that cytokines, a number of cytokines, but here I'm pointing out IL-31, can actually directly stimulate the sensory nervous system, much like histamine, and can evoke itch even when activating only a small population of sensory neurons within the dorsal root ganglia. And indeed, blockade of IL-31 is emerging as a potential therapeutic strategy in the context of atopic dermatitis associated itch. So in terms of our work, based on these phenomena, we actually asked a very simple question. If a cytokine like IL-31 could stimulate the sensory nervous system, and with the identification of these innate cell populations that are rich sources of these canonical type 2 cytokines, could we modulate itch? Could these cytokines directly modulate itch in a similar manner? To test this, we simply took a very simple approach. We extracted dorsal root ganglia from both mice as well as cadaveric human donors, and we looked for expression of type 2 cytokine receptors, including IL-4 receptor alpha, which signals both the IL-4 and 13, which I'll focus on mostly here, as well as the native IL-13 receptor, and as you'd expect, the IL-31 receptor, and this was conserved between both mice and humans. We could actually never detect the IL-5 receptor from the dorsal root ganglia. So understanding now that the receptors are actually expressed within the nervous system, we then sought to test whether these cytokines actually have functionality. So do these canonical cytokines actually activate the sensory nervous system? To test this, we take a number of different approaches. One simple approach is to simply extract sensory ganglia, label them with a dye that turns red in response to calcium influx, and we can then detect activation. Before we put on the IL-4 into the petri dish, we have nice plump green neurons which within a couple minutes become activated, as we can detect by their change in color. So then we wanted to test whether this is not just functional in the petri dish, but important in terms of a disease model. To do this, we used a well-established model of atopic dermatitis-like disease in which mice are treated with the irritant MC903, which is calcipotriol, or many of us know as Dovanex. This very reliably induces AD-like disease in vivo, along with both clinical histologic as well as immunologic features characteristic of atopic dermatitis. This model was pioneered by Mei Li when she was with Pierre Chambon. So we took a very simple approach. We just deleted the IL-4 receptor from the peripheral sensory neuron. We engineered mice. The receptor was intact everywhere else in the mouse, but deleted in the peripheral sensory neurons, and we subjected these mice to AD-like disease. And when we did this, we were able to identify that there was a marked and significant reduction in terms of itch, shown on the left, over time, as measured by scratching bouts. Additionally, we saw a significant marked reduction in terms of the clinical parameters as well, other clinical parameters, including skin thickness as well as histopathologic inflammation. So in the interest of time, and given that this has been published for a little bit, I won't talk about the data, but we think that this informs in part how drugs like Dupilumab that block IL-4 receptor alpha may function to limit itch. We think that this works not simply by blocking a pruritogenic effect, but rather these type 2 cytokines seem to promote neural hypersensitivity to a variety of other pruritogens. This makes a lot of sense to us because in the setting of lesional atopic dermatitis skin, so many, actually even known pruritogens are upregulated in that context. But at the same time, we were interested in more broadly and more deeply understanding how itch at the neuronal level is triggered. We know that a number of cytokines in lymphocytes have to go through the JAK-STAT signaling pathway by way of their receptors. These are just a handful of cytokines that go through the JAK-STAT signaling pathway. Many, many more do. And so we thought perhaps by somehow getting at JAK, we can actually choke off pruritogenic signals simultaneously from a variety of different sources, perhaps even beyond IL-4 and 13. To test this, we took a very similar in vivo approach. We used our atopic dermatitis-like disease model. We generated mice in which JAK1 specifically was deleted in the peripheral sensory nervous system, and we looked to see if there was actually a reduction in itch in the context of disease. In fact, there was. Even when there was no difference if we looked very early in terms of inflammation. This was an important concept for us because up to this point, the kind of central dogma in treating atopic dermatitis was that we really just have to immunosuppress and get rid of the inflammation. But we were really playing with this idea that we can now dissect out specific itch mediators even in this complex inflammatory environment to get at itch apart from inflammation. And this is a central theme in our laboratory right now. It's just really understanding the components that drive itch. So we know that JAK inhibitors on the market actually have anti-inflammatory properties by acting on a variety of immune cell populations. But what we're proposing and exploring now is the idea that these JAK inhibitors may actually have neuromodulatory properties given that these neurons actually respond to a variety of cytokines. The reason why we're interested in this is that we see a number of patients from all across the country who come in with severe chronic itch. The problem is they don't have atopic dermatitis, so they can't simply get Dupilumab. They have chronic pruritus of unknown origin or chronic idiopathic pruritus, so they have itch without a rash. Many of these patients actually have no effective treatments. They're highly refractory to even anti-inflammatory therapies. And in our clinic, on average, these patients have itch that is more severe than even our moderate, leaning on to more severe, moderate to severe atopic dermatitis patients, and shown on the right. 0 on the scale being no itch, and 10 being the worst imaginable itch. If we actually biopsy the skin from these individuals, it has been hypothesized by many of the clinicians I listed earlier that these patients actually have a form of immunosenescence that results in an allergic type low-grade inflammation of the skin. We sought to test this by simply performing RNA sequencing of their skin, and what we found is that no question their skin is distinct from control skin, as is atopic dermatitis, but they seem to cluster more closely with atopic dermatitis lesional skin. They remain distinct, but despite the fact there's no rash, there seems to be some level of low-grade inflammation. Putting this all together, we were able to get JAK inhibitors for a small cohort of patients, all of whom responded, some quite exquisitely. Because of the level of inflammation that these patients are known to have, this low-grade inflammation, we do at times give broad immunosuppression, such as cyclosporin. Even when that does not work, when we switch over to the JAK inhibitor tofacitinib, these patients do respond quite nicely. More patients have been able to receive this drug since this time, but this is what we have published thus far. Bringing this back to atopic dermatitis, topical JAK inhibitors have actually been employed for atopic dermatitis, and they've been shown to be quite effective. What's unique is that they've also demonstrated very potent anti-itch properties. We were fortunate early on to share some of our work with Incyte Corporation, with whom we were able to actually form a collaboration and design a phase 2 randomized trial with their ruxolitinib cream. We were actually able to design a trial where 307 patients were enrolled across 6 different groups. They were randomized into either vehicle, active control, which is triamcinolone cream, essentially standard of care, and a variety of different doses of ruxolitinib across a wide range. Patients received treatment for 8 weeks. By the end, were able to opt into a open label period for an additional 4 weeks at the highest dose. In terms of efficacy, improvement in terms of their disease severity is measured by EASI on the y-axis. There was no doubt an effect of the active control as one would expect, triamcinolone cream. However, there was also very much a dose-dependent and time-dependent effect, increasingly with each increasing dose and over time of the ruxolitinib cream. And we see that there's an increasing dose response as well as a time response and a maximum response at the highest dose there. In terms of itch, what we found was that there was a significant and rapid improvement in itch, particularly at the highest doses of ruxolitinib, that was sustained over time. And we can compare this actually to triamcinolone cream shown here in the gray curve. So also recently, the first patients with germline JAK1 gain-of-function mutations were identified. These patients presumably have JAK1 activated in all of their cells in their body. Unfortunately for these children, they have very severe inflammation in the skin as well as itch. And interestingly, when these patients actually receive very high-dose steroids to treat the inflammation, their itch actually persists. And it's only after they actually, through compassionate use of ruxolitinib, once they were identified to JAK1 gain-of-function mutations, were able to actually sleep for the first time throughout the whole night, and actually their itch was actually responsive. So we're speculating that these individuals may actually have neuronal itch just driven by their JAK1 gain-of-function mutations. So starting with the identification of epithelial cell-derived cytokines and looking downstream of these events and trying to understand how the epithelial barrier actually regulates inflammation, we've actually stumbled on a number of different pathways, both in the immune and nervous systems, in terms of how this type 2 axis actually contributes to itch, particularly in a context of atopic dermatitis. I want to bring you back to the mast cell and histamine here. So we know the pathway that's been most well understood over time has been how histamine induces itch, and that's why I brought it up in the beginning. And the way that the histamine is most associated with the mast cell, often in tissue resident, and in the setting of atopic disease, they're coated with IgE, they bind antigenic components of allergen, and then they degranulate and release histamine. And as I mentioned earlier, histamine can tickle the nerve. But what's been known since the 1950s is that a compound called 4880, shown here, can actually activate mast cells, and it can make mast cells release some histamine. And it's also been shown that this compound, when it's cyclized to a tetrahydroisoquinoline, THIQ motif shown here, that not only is this motif shared by a variety of different peptides, both endogenous as well as drug peptides, exogenous peptides that have been developed, that it's much more potent with this structure in terms of activating the mast cell. And it turns out that the mast cell is actually activated by way of a G protein coupled receptor called MRGPRB2. and discovered by Xinjiang Dong. What's interesting about this receptor is that it's in a family, a whole family, many of which are present on the sensory nervous system, but this one in particular is specific to the mast cell. So the next part, we've actually collaborated very closely with the Dong lab and explored if there is this alternative pathway for mast cell activation, is there a different kind of itch that can be evoked in contrast to this classic histaminergic itch? And in fact, as I've alluded to already, this THIQ motif is conserved across a variety of different compounds, as well as endogenous molecules such as proagenomedulin peptide. This peptide is actually produced mainly in the adrenal medulla, but it's also been reported that this peptide is produced by keratinocytes. And this is, again, an endogenous peptide. So we then simply tested whether this peptide could actually evoke itch in this kind of manner, but in a way that's dependent on MRGPRB2 and dependent on mast cells. And to do this, we simply injected in PAMP, the PAMP peptide, into both wild type as well as MRGPRB2 knockout mice. And what we found was that, in fact, was that there was a dose-dependent response, and that was largely abrogated in the setting of MRGPRB2 deficiency. Further emphasizing the point about potentially histamine independence, this pathway appears to be completely resistant to any treatment with antihistamines as well. And interestingly, what we can do is actually do in vivo calcium imaging. So what I showed before was in a petri dish, but we could actually do this in vivo. And we could look at how neurons are activated in response to a stimulus. So if we actually inject PAMP into the skin, we see that at baseline there's no response, but then in the lower panel, when it's activated, we see this, the neurons lighting up. In the interest of time, I won't show the data, but the pattern of activation is quite distinct in response to PAMP, in contrast to IgE and the histamine pathway. So there's a spatiotemporal pattern that is quite distinct, indicating again perhaps that this actually is a different kind of mast cell mediated itch. So it's been actually reported that mast cells are enriched in the setting of allergic contact dermatitis. So I want to make a distinction between atopic dermatitis and allergic contact dermatitis. And in fact, that's what we found by avidin staining shown on the top. And there's increased PAMP peptide staining within the epidermis, in fact, in lesional allergic contact dermatitis skin. So then the question is, can we model this as we've done even with our other atopic dermatitis experiments? And there are a variety of different models of allergic contact dermatitis, and if we subject MRGPRB2 knockout mice to these different models, including ascorbic acid, oxazolone, and DNCB, we see that there's a reduction in terms of the allergic contact dermatitis, hapten-associated itch in the setting in vivo as well. So we think that in the setting of allergic contact dermatitis, there are endogenous peptides like PAMP that can be upregulated. This will activate MRGPRB2 and can activate alternative effector mechanisms beyond the classical IgE histamine axis to evoke itch. And this may actually be an important mechanism of itch in the context of allergic contact dermatitis. As an immunologist, we always like to divide hypersensitivity reactions. This is essentially what you read out of an Immunology 101 textbook. We have classical type 1 IgE mast cell activation, as well as type 4 delayed type hypersensitivity, which underlies allergic contact dermatitis. We know that part of this paradigm can explain certain kinds of itch, like the classic histaminergic itch. However, in large part, we can't, through these paradigms, explain a lot of forms of inflammatory itch. So although we have known for decades that type 4 hypersensitivity underlies allergic contact dermatitis, what I'm proposing is that it's hard to understand itch in the context of this immunologic paradigm, as I'm showing you here today, that MRGPRB2 and mast cells may actually be very much involved. So I think there's an increasing understanding of how the neuroimmune interface is quite complex. There are a variety of different immune cells and molecular mediators that interface at this synapse of sorts. And what I think is going to happen is that this is really just the tip of the iceberg, and there's a lot more to be known about these various different kinds of interactions. Furthermore, I'd like to also say that although we're interested in itch, we feel that itch actually represents a broader neurosensory paradigm that can affect many, many organs throughout the body as well. And we've been studying atopic dermatitis for a long time, and we think that this has been able to inform even things like genetic disorders. We've been able to identify various cell populations and molecular therapeutic targets both in the immune and nervous systems. And what's been very exciting for us is that this has been able to inform one of the most active therapeutic development phases in dermatology that also extends to a variety of other clinical disorders as well. I'd also like to just end by saying that we're also interested in a number of individuals have really advanced our understanding of how neuropeptides actually from the sensory nervous system modulate inflammation at the barrier. So in addition to understanding how cytokines and various inflammatory factors affect the nervous system, we're also interested in understanding this entire circuit. So finally, I'd just like to thank the talented individuals in my laboratory, our wonderful collaborators, both at WashU and elsewhere, the Insight team with regard to the clinical trial, Xinzong Dong, which we did jointly with the MRG-PRB2 studies, our funding agencies. And today I'd really like to especially thank Steve Katz and my many mentors, David Artis, Lynn Cornelius, Wayne Yokoyama, and John Stanley, who've done so much for me over the years. I'm really grateful for them, and I wanted to publicly acknowledge their support. I'd like to thank our patients. They've been very, very supportive of our work, and I'm incredibly grateful to them as well. And thank you all.